This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 103, recorded on April 30th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. This episode is sponsored by ASM and the International Society of Chemotherapy. They're joining together for ICAC ICC 2015 this September. This will be the principal meeting for clinical microbiologists, infectious disease physicians, researchers, and pharmacists to talk about antibiotic resistance, new antibiotic development, and antibiotic stewardship. If you go to this meeting, you'll be able to choose from over 110 sessions and 19 courses that explore major advances in drug development and research on infectious diseases. You can earn up to 32 3/4 hours of continuing education and network with more than 6,000 professionals who are working to improve diagnosis, prevention, and treatment of infectious diseases everywhere. Plus, you'll be able to explore the latest products and services from over 70 exhibiting companies. Abstract submission and registration are open through May 21st. Go to icac.org/ICAC-ICC for meeting details. Joining me today from Small Things Considered, Elio Schaechter.
Well, hi. How are you doing?
I'm well. How are you?
Very well, very well.
We're finally out of winter.
Oh, good. We can't brag about the weather in San Diego anymore.
Well, you could. It's probably better than here, but it's really nice. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
Michele just ran from the golf course.
It's not true.
I know. I'm kidding. But you have a good sense of humor, so you're kiddable.
I sure do.
But you would have liked to come from the golf course, right?
I would have.
Of course, if you were there, you wouldn't come to do TWiM, though. You'd just keep playing.
No, I would. TWiM is a priority. It's a pleasure to be with you all.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Chamber of Commerce.
It is today. Yesterday, we had the monsoon.
How many years have you been in Charleston?
This is my 26th year.
And you're like 32, right?
Absolutely.
Wow, that's a long time.
What a lovely city you're in.
I've never been there.
Oh,
Well, we'll have to bring you down when Dr. Trackman comes with her van from Milwaukee, and we've recruited Vincent's old colleague, Dr. Trackman, the immediate past president of the American Society for Virology, and she's going to head up our new graduate school, and she's going to join my department.
Congratulations.
You have a new graduate school?
No, she's our new graduate
dean. Okay.
She's our new dean.
Michael, when she gets there, she won't want me to come, so you might want to get me before.
Okay.
The food is
she
great
knows you.
in
She
Charleston.
knows you. That's the problem.
Yeah. Oh, we were postdocs together. Actually, she was a graduate student with David Baltimore. I was a postdoc, and we knew each other back then. This is like over 35 years ago. The food is good in Charleston. Is that what you said?
Fantastic.
It's off the chart.
Really?
Off the chart.
Hey, I'd move there, but
crab soup. I know.
I'd move there. I'd retire there, but I wouldn't retire. I'd go there to podcast and blog and teach.
Oh, yeah.
But,
What time? What time are we moving, Vincent?
You want to move
Count me
too?
in.
You can leave behind your snow shovels. When I moved out of my postdoc in New York to Charleston, I left the snow shovel behind and have never regretted it, except when I went home to Chicago to shovel my father's sidewalk in the wintertime when I would be visiting at Christmas time.
Michele, we could do TWiM in person.
Yeah, I'm gonna have your people talk to my people,
Right.
and I'll continue working on a book with Elio.
Yeah. Maybe we should all go to San Diego, but there's no water
Hey,
there.
that's not bad.
All right, today we have a paper and a snippet. We're back to snippets, and Elio has that for us.
Okay, my snippet is a little bit different than the usual because it deals with protozoa, protists. The general subject is that if you look at protists anatomically, you find a diversity of forms and shapes in a single cell that boggles the mind. Even consider paramecium. Paramecium has a mouth, a gullet, an anus. It's called a cytoproct, polite society. Something like a bladder. Food inclusions. It has, besides cilia and all that, it has a morphological variety that's staggering. And you wonder, why is this a unicellular being? And I don't have the answer to that, except to say that way back when multicellular organisms arose, some of the unicellular organisms went their own way and kept on evolving. So there are sort of two ways of achieving biological complexity. The multicellular one is the most popular one and the most successful one, but that doesn't mean that the unicellular, monocellular one didn't go a long way. So let me tell you about a structure called the ocelloid, O-C-E-L-L-O-I-D. The ocelloid is an equivalent, I kid you not, of the eye of vertebrates, okay? This is in a unicellular dinoflagellate. Not all dinoflagellates have this structure. It is unique to a group of dinoflagellates called the varnovids, in case you want to know. They are marine forms. Okay. Now, let me tell you about the ocelloid and why it's so awesome. In fact, a blogger that had a blog called the ocelloid called it the awesomest thing in biology, and I think that's very accurate. I can't think of anything awesomer. Let me give you the details anatomically. This is a 20 micrometer in diameter structure that looks like an eye, like a mammalian eye. It's only one 1/1000th the size of a human eye, okay? And it has all the details. It has, get this, be ready, a cornea, a lens, an iris, and a retina, equivalents, okay? And if you cut through it and you compare it to a vertebrate eye, they look almost the same. This is in a unicellular organism. Now, you agree this is awesome, okay?
How's that possible?
Evolution.
Well, it's possible this way. The business end of this organ, let's call it, is a retina-like body. And the retina, I'll jump into that, is essentially a modified chloroplast. It has, or a cyanobacterium, if you wish. It has thylakoids, which are these tubular membrane structures, which are characteristic of photosynthetic entities, and it is not doing photosynthesis. In fact, the whole organism is not photosynthetic. It does not have a chloroplast. But it has a modification of a chloroplast which detects light. What it does with the light is not clear. I'll hasten to say. The whole thing is having an eye in this case did not evolve for the purposes of reading the New York Times, I don't think. Okay, so we don't know why exactly, except that these dinoflagellates live in dim light, and this is a way of enlarging light, because the lens is a refractive lens, a lot like ours, which gathers light and shines it onto this retinal body. Okay, so that's the story, and it is totally unbelievable. Whenever you read the literature, and you find words like stunningly complex and amazingly, etc. It lends itself to superlatives, and for good reasons. Okay, so I'll tell you a little bit about the paper. The paper is by a group of people from various countries: Japan, Switzerland, Taiwan, and Saudi Arabia. And the authors are Hayakawa, Takaku, Wang, Horiguchi, Suga, Gehring, Ikeo, and Gojobori. Quite a group, okay? Anyhow, what they did is essentially a fairly thorough anatomical study and functional study of the ocelloid. So they go into the retinal body. Let's call it the retina. What the heck? I mean, they like to call it retinal body because it's for obvious reasons, but I'll call it the retina. It has this set of parallel lamellae which change in thickness whether there's light or dark. So there's a an atomic modification of the structures depending on the light. And just what's involved in that is not clear, except that somewhere along the line, actin participates. And the changes in morphology of the retina are inhibited by cytochalasin B, which is an inhibitor of actin polymerization. So actin somehow is involved in the response to light. You don't find actin in the lens. So the lens probably is not like our lens. It doesn't change shape. It's just there to in a certain geometric position. By the way, the eye, this eye-like structure is so complex that, and so much like a vertebrate eye, that I wonder if in old age these dinoflagellates don't get cataracts. Okay,
it really looks like a science fiction eye, from a bad science fiction movie.
Right. It's amazing, really. Anyhow, so they studied the variation in shape and form with the light. So there are a couple of questions. So this is understood and emphasizes the structural and possibly functional properties of this eye. The organisms, as I say, live in dim light, and maybe this is a way to perceive light. Now, for what purpose they need light is just not known. And I say the two big unknowns are why they have this, what is the functional meaning of it, and the other is how do they transduce the signal from light to whatever it is. So both things are related. Those are two questions which are outstanding. And I would hope that somebody picks this up. Well, I imagine people are going to pick this up and solve this particular question. I'm not even sure you can culture this dinoflagellate. You can culture a number of dinoflagellates, I think. So this should be all right. One other thing which is fun is that in the retina is a rhodopsin-like gene. So it is, and it turns out to be a bacterial type of rhodopsin. And so that and the other properties of this body let you call it an organelle. It has DNA, by the way, and the DNA, curiously, has not yet been sequenced. But it'd be very surprising if it didn't turn out to be related to bacterial DNA. So here is so this whole thing was probably acquired by horizontal gene transfer. In other words, there was a photosynthesis. Let's make a case for this. I don't think anybody has seriously said that, but let's pretend we know that a photosynthetic bacterium, maybe a cyanobacterium, got swallowed by the original cell, and instead of becoming a chloroplast, it became an eye. That's more or less what that's consistent with what we know.
Yeah, the genome is gone now, right?
The genome is more largely done. Yeah, but as I say, we don't know very much about the DNA. I can't even tell you how much DNA there is. Maybe it's in the paper, but I didn't catch that. Anyhow, it's possible that this is how there was a distinct evolutionary bifurcation of a cyanobacterium going one way to become a chloroplast, another way to become an ocelloid. How do you like them beans?
It's pretty cool. According to this paper, Elio, and the methods, they collect these samples,
right,
in a bay.
But I think that maybe there's no reason why they can't be cultivated. So it's just a question that now then it's not a well-developed system. Very little work has been done on it, and so you do the easy things first, and time they'll work this up. So stay tuned because the ocelloid is with us.
If you search PubMed for ocelloid, you get 4 papers.
Yeah.
I don't know. Maybe PubMed is not the place, right? Because it's mostly life.
It's probably not indexed there. Yeah. It's probably not indexed there.
But
though the PLOS journals are indexed in PubMed, so
yeah, no, it's a good indication that very little work has been done on it. In the old days, the morphologists of protists did a lot of work. But as I say, the most general message is that there is a alternative to multicellularity when it comes to biological complexity.
Yeah, it's cool. It's very cool.
Okay.
There used to be a blog on ocelloid, and she stopped writing.
That's right. She stopped. That's right. And in the blog, she calls the ocelloid itself, homonym, the awesomest thing in biology, as I said.
That's cool.
And as I said, I can't imagine anything awesomer.
Viruses are pretty awesome.
They are, but
Still,
man,
it's
this
so
takes
elegant.
the cake.
Viruses are pretty elegant. They don't play golf, but they're really elegant. I can understand. This is the fact that it's a single cell is pretty amazing, but the fact is that awesome is in the eye of the beholder.
I was going to say, I'm not sure
Oh,
we're going to see eye to
Vincent.
eye on this one. Vincent.
Normally, it's Michele who gets those puns in.
She does?
Yeah.
Oh.
She's been known to.
Yeah, I don't do many, but when I do, watch out.
Yes.
Oh, yeah, they're lousy.
Yeah. They are lousy. What can I say? Italians
Anyhow,
are not known.
we're writing this up for Small Things Considered, so you're going to see it there as well.
Nice. Thank you, Elio. Nice little snippet. I love the name. Ocelloid is terrific.
It comes from ocellus, which is a kind of eye, a word for eye, from os, somewhere in the root is something related to eye.
Okay. This week's episode is sponsored by SciMed Solutions, a leader in cutting-edge software products in science and medicine. From EKG management to sequence analysis, the software that's built by SciMed helps researchers and organizations increase their throughput by orders of magnitude. That's 10, 100, 1,000-fold. This allows them to get results more accurately and efficiently. SciMed has years of experience working with scientists in immunology, virology, and genetics. If you are going to this year's conference for the American Association of Immunologists in New Orleans, stop by and say hello. They're in the exhibition hall, and they can tell you all about what SciMed can do for you, and you can see their new product designed for life scientists. SciMed software and consulting services will let you automate repetitive analysis and reporting of large data sets. You can streamline experiments and maintain data integrity. Contact SciMed about research partnerships, database setup, mobile apps, and web applications. Cool. Find out more at SciMedSolutions.com. SciMed Solutions, innovative software empowering the world's leading minds. All right, I have a paper for everyone, which is really cool. It's pretty awesome, I think.
It is.
Not as awesome as an ocelloid, but still pretty awesome. And this is by Matthew Barber and Nels Eld, and it's been published in Science in December. And the title is Escape from Bacterial Iron Piracy Through Rapid Evolution of Transferrin. Now, I should point out that Nels has also done some work on viruses. And in fact, he has been on TWiM. Couple of years ago at one of the ASM general meetings. We did a live TWiM, and Nels was on that. Talked about some of his virus
was Matthew as a postdoc who decided to take a lot of the principles and approaches that Nels has developed to apply them to this really interesting bacterial interaction that you're going to tell us about.
You know, it's funny. Nels said he wants to make the TWiFecta. He wants to be on TWiM, TWiM, and TWiP.
Speaking of puns.
So I suspect they're going to do it. Well, you'll see in a moment. This could also be done with parasites. So this is all about iron, which we have talked about here on TWiM before, the battle for iron. It's an essential nutrient for not only us and other animals, but microbes as well. So, we have battles over iron. In us, it's in heme, right? It's a big deal there. In our blood can be incorporated into iron-sulfur clusters or redox cofactors that you would find in metalloenzymes. So it contributes a lot. And apparently at any time we have 4 to 5 grams of iron in our body.
4 to 5, did you say?
4 to 5 grams. Yep. It's amazing we don't set off the metal detectors. I guess that's not enough.
Probably not concentrated enough.
Yeah, but I guess if you ate too much, it might. Oh, no, I'm just kidding. Anyway, so we get a lot of our iron by eating foods with iron in it. It's absorbed in the intestine, and then it's bound up by ferritin, a protein which binds iron. And when cells need it, it's taken into the cell, bound to ferritin, by receptor-mediated endocytosis, and as the endosomes go in the cell and they acidify, the iron is released, and that goes into the cell for its use, and then the ferritin is put back on the surface.
Let's be cognizant that it doesn't only go into hemoglobin, but in many cytochromes.
Oh, many other
enzymes.
Yep, iron sulfur clusters, metalloenzymes for sure. And so we have a system for binding it, ferritin, and there's a ferritin receptor on the cell surface that takes it up and so forth. So this paper is all about ferritin. So the microbes want to have iron, and so they have a lot of tricks to get it. And sometimes we've talked about some of those.
Yeah,
here on
the siderophores.
The siderophores, they have a huge affinity for iron, so they grab it away from whatever's holding it, and we try and counter that as well. So if we get infected, we have inducible mechanisms for sequestering iron. So it's a real battle for iron to see who is going to win. There's actually a terrific review article. It's called Iron in Infection and Immunity, and I'll put a link to it in the show notes. It just talks about why we need iron and why bacteria need it, and the fight, basically. It's really good. So,
who wrote that?
James Cassatt and Eric P. Sakar, and they're from the Department of Pediatrics at Vanderbilt University Medical Center in Nashville.
Wow. It was just there. Yep. It's in Cell Host and Microbe. Really, really good review. So this is when we try to sequester iron so microbes can't get it. This is called nutritional immunity. It's actually a new word for me. I hadn't known that this, and it's in the article as well, and in the science paper they talk about it. Just ways to sequester iron so that bacteria can't get it, and the ways bacteria try and get around it. Really quite interesting stuff. And it's not just bacteria that want our iron, right? Plasmodium falciparum and the other malaria parasites, they go inside red blood cells to steal it, right? So this is a really important metal, and a lot of people want it, and a lot of microbes want it. So here, the question is ferritin, which is this iron-binding protein. It is a interesting protein that has an N and a C lobe. These are two identical lobes that have duplicated years ago. So originally it was one lobe, a protein consisting of one lobe. It duplicated, so now it can bind two molecules of iron. And this ferritin-binding protein, as I said, is in us that carries iron around, because you don't want iron by itself floating around. It has to be bound to ferritin. So the question is, has ferritin evolved over the years driven by pathogens who try to steal iron from it? And this is actually, this kind of host pathogen conflict at the level of evolution is a brand new field developed by people like Mike Emmerman and Harmit Malik, and then Harmit trained Sarah Sawyer, who is now in Texas, and she continues to do this with viruses. And I believe Nels worked with Sarah Sawyer,
Nels
right?
was also a postdoc.
So now Nels is continuing this on his own. And so Sarah...
They focused on viruses primarily, right?
That's right. So both Harmit and Sarah Sawyer and Nels, they've all been on TWiM to talk about their virus work. And the way this works is, so you take a viral defense protein that exists in a cell. So we know what they are, and we look at the phylogenomics of it. We get different genes, say a protein like PKR, which is an innate defense protein, and you just get the gene from different organisms along the phylogenetic tree, and you sequence it, and you ask, are there any amino acids that seem to be positively selected over time? In other words, they've been subjected to natural selection, and a change has been fixed in the population compared to other surrounding residues, which don't change as much. So we look for positive selection. And then you can take those proteins and ask what the different alleles, if you have different amino acids at a positively selected residue, you ask, how do they fare with the virus protein that they're interacting with? And so these individuals have done lovely work to look at this. Sarah Sawyer, in particular, we did a story of hers where two different viruses bind to the transferrin receptor, and they could see positive selection in the receptor over the years, and they took those changes and showed that they interfere with virus binding. And the cool part is they don't occur in the place where ferritin binds to the ferritin receptor. So you can look for positively selected residues in a protein. Then you can test to see if they're functionally important, and that's been done for viruses. So here, Nels and Matthew are doing it with bacteria. Now, they point out at the beginning that ferritin is a tricky one because, it may be subject to bacterial pressure, but it's also got an essential host function. So can it change and still preserve that function? But, I've just told you that his boss, Sarah Sawyer, has done this with the transferrin receptor, which, of course, also has a host function and binds at least 2 different viruses. So there's a precedent for that. So what they did here is they cloned...
Having 2, being a duplex molecule helps here.
Yeah, they have a kind of an internal control, which is kind of cool.
Having a duplex certainly is one way around it. And but molecules have a lot of surfaces, right? So one surface can do one thing, and so I don't have any problem with that unless the surfaces are completely overlapping. Then you have an issue. So what they did here is they cloned and sequenced transferrin genes from 21 hominid, New World, and Old World monkeys.
And apes.
Is that part of the hominid?
Yeah.
Yeah. Okay. Hominid, New World. No,
I don't know the exact definition of hominids.
Well, that's what gorillas, chimpanzees,
I think that's
included.
That's included.
Apes. I think we're apes.
Yeah, I think they throw them in with the hominid, right?
Okay.
And if you look at these amino acid sequences, there's lots of conserved regions, but you can apply bioinformatic analysis to them and find some evidence of positive selection. And remember, these are phylogenetically related. You can make a tree according to the evolution of these animals, and then you can see how's the protein changed over time. So what does it look like? Ferritin, remember, has 2 lobes, N and C. They find 16 out of 18 rapidly evolving amino acid positions map to the C lobe. Even though they're both the same functionally, apparently, they both bind a molecule of iron, most of the rapidly changing sites are in the C lobe.
And that's not going to happen by chance, right? Statistically, that's significant that so many of them are in the C, but not the other lobe.
Not likely, right? If they're doing the same thing, right?
So that was a great clue.
Yeah, these guys are really... I guess you look and you're going to find things, but this is a lot of cool things that happen in this story. So that was the first thing. They probably got all excited when they found this. Right? So as an example, in the hominid or hominoid lineage, there was no selection at all in the N lobe. So it's all in the C lobe. So they've been subjected to different kinds of selection pressures. Right away, you can tell that. So one of the questions they could address right away is, we know that the transferrin receptor, of course, which binds transferrin, has been subjected to positive selection by viruses. Remember, I told you at least 2 different kinds of viruses bind to it as a receptor, and they could show that the sites that change in transferrin are not those that contact the receptor. So you could say it's the receptor that's driving these changes, but that's not the case. And that's because this previous study by Sarah Sawyer's lab. Now, a number of bacteria that are pathogenic for us, like Neisseria and Haemophilus flu, have on their surfaces receptors whose function is to grab iron from transferrin. And these receptors or proteins are called transferrin-binding proteins, and there are different kinds. And the one we're going to look at here is A, transferrin-binding protein A. So it's a transmembrane transporter. They have a lovely picture. Unfortunately, this will probably be behind a paywall, although Nels has a great website. He might have a picture there. They have a beautiful picture of this transferrin-binding protein, which kind of looks like a basket in the outer membrane, and then it's binding transferrin. It just wants to rip away the iron, and then it's going to put it into the bacteria. It's so nasty, Transferrin
that
binding.
art was done by Janet Awasa.
Mm-hmm.
They credit on their blog. It's beautiful.
Is there a copy of that on the blog?
Well, Matt Barber, the postdoc who was the first author on this work, has his own website, and it's there.
Oh, good. People can see it. We'll put a link to that.
Yeah, I'll link to his website.
It's a really pretty picture, and you can see right away the iron there, and then this bacterial protein is trying to grab the iron. Okay, so the point is that this transferrin-binding protein binds transferrin, and so you could say, well, are these sites on transferrin that are evolving? Are they contacting it? Well, it turns out that recently the crystal structure of human transferrin bound to the transferrin-binding protein A has been solved, so they can take their evolving sites and ask, are they at the interface of the two proteins? And indeed, 14 out of 16 of these rapidly evolving sites on the C lobe directly contact the transferrin-binding protein. Very cool. And also there's another transferrin-binding protein called TBPC. It binds the C lobe. No evolving residues are contacting C. So the ones in transferrin are contacting transferrin-binding protein A. Okay, so far we have a bioinformatic result, which is telling us that there's some amino acids on transferrin that with time in different species are changing, and they seem to be contacting transferrin-binding protein. So now they do some experiments. They do some wet experiments, as we say, and they're very cool. They're going to do competitive binding assays to look at the interaction between transferrin and this transferrin-binding protein and ask if the amino acid changes make any difference. So they produce transferrin from a variety of species. They produce it from cloned DNA and they purify them. And then they put TBPA from Neisseria gonorrhea and Haemophilus influenzae. They produce these in E. coli separately. E. coli doesn't have these transferrin binding proteins.
Thank goodness, otherwise we'd all be dead probably.
Yeah, right.
I wonder if it's true for all E. coli.
Well, it's interesting when Vincent commented on the nutritional immunity concept. I was racking my brains trying to remember the first time I heard it, and as I stumbled through the paper, the first reference was from Gene Weinberg, who was on my thesis committee at Indiana. And he is the originator of nutritional immunity.
Cool.
So that's
really,
yeah, and it's in a JAMA paper from 1975. But I think where Vincent is going to take us next is this concept of the Red Queen hypothesis, where the warring parties, not only does one species have to continue on its path to become better, but it has to do it while things are trying to constantly kill it.
Yeah, that's what this...
And that's what these cool experiments are going to do for us.
Yeah, these are all the ones I cited before. They're, one way to characterize them is host virus conflicts, but the other is Red Queen conflicts, because both sides change back and forth, but no one gets anywhere.
Right.
You end up being in the same place, which is what the Red Queen does, right?
Right.
Michael, if you look at the gut microbiome, the genome, and you search it for transferrin-binding proteins, will you find any?
Yes. I'll bet you they're in the Clostridia.
Mm-hmm.
And
so part of what makes them problematic is that they're taking iron, right?
Yeah. And that's a good experiment to do, because we can now do that experiment with, and there's other anaerobes in our gut that also similarly have these things.
Yeah.
E. coli tends to just use siderophores rather than going after transferrin, which is probably not abundant in the gut.
Mm-hmm. Yeah, so there are other mechanisms of stealing iron.
Yeah, but on the mucosal surfaces, then
right.
It's a different strategy.
All right, so back to this interaction assay. They have these E. coli producing TBPA from Neisseria and Haemophilus. They take the E. coli, the whole cells, they spot them on a membrane, and then they add human transferrin, which is conjugated to horseradish peroxidase, and then they add different amounts of the primate transferrin. This is a competitive assay, right? And the primate transferrin doesn't have horseradish on it, so you're going to see if that competes the binding of horseradish conjugated human transferrin to the E. coli, which is producing the transferrin binding protein. Okay. They then detect human transferrin by chemiluminescence. They add a compound that is broken by HRP to form luminescence. Okay, so it's a very simple assay, and they have these pictures of the dots, which they can show you, and you can see what's going on. So the results are that both of these pathogens, and Neisseria and Haemophilus, the TBPA binds human transferrin. Okay, quite clear. They also bind transferrin from gorillas, but not chimps, not orangutans, not gibbons, and not baboons.
Yeah, they make a point that the gorilla connection is kind of weird because you'd expect us to be closer to the chimps than to gorillas, and this doesn't look that way.
It doesn't really. But
it's a detail. I don't think.
Okay. And they confirm these binding with another assay, an ELISA assay, but they're correct. So chimp and human are different in this binding assay. They differ by 4 amino acids, and one of them, it's at position 591. It's glutamic acid in human, a lysine in chimp, is one of the positively selected residues that they identified, and it's near part of when TF interacts with TBPA, it's near the interaction site. It's important for iron acquisition. So if you take the human transferrin, which has glutamic acid, you change it to a lysine, make it like chimp at that position. Now it doesn't bind TPA from both of the TBPA from these pathogens. And conversely, if you put glutamic acid into the chimp, you change lysine to glutamic acid at 591, it will now bind TBPA.
So I have one question about that because I was concerned about the chimp story and why it was so different than us. Is it because there are so far fewer chimps that are adding to the gene pool and the bacteria having a shorter generation time, and there are so many more humans that the microbes are adapting to the bigger population rather than the smaller population? I may not be saying that in the right way from a bioinformatic perspective, but we know that the gorilla populations are, dangerously low, and the chimp population is dangerously low. So as we're thinking about, this, if you will, Red Queen hypothesis where you're interacting back and forth, do you need what's the critical population you need in order to see the effect?
Well, chimps.
Interesting point.
Chimps weren't always low numbers.
No.
So that I don't know how old the change is.
Yeah, and that's the one question I don't know how to ask bioinformatically. As I'm reading casually a science paper to figure out.
It's hard to know because all you know is chimp. And you don't know when, and they've been around a long time, so you don't know when. Bonobos have a glutamic acid at this position, which is the human sequence. So they say this indicates that the change is recent because bonobos are more ancient than chimps, I presume. But I don't think we know. I think that could be, but right now there are like 1.5 million chimps left in Central Africa only, in zoos, of course, but. At one point, there were many, many more, so I'm not sure that numbers plays a role. It depends when the change got fixed, and I don't know.
Yeah.
You know that. Anyway, so this really provides some evidence that this interaction and these positively selected residues in transferrin they could limit the host range, right, of these pathogens. If this is if this completely deals with iron acquisition, I mean, there may be other ways that it's acquired, so this may not be the whole story. But
there's a parallel story about the host range with the battle for iron. Eric Scarr, Vanderbilt, who wrote that review that you mentioned, they have been studying staph, and one of the problems with studying staph pathogenesis is it won't grow in mice, but it grows well in humans, and it turns out the difference is maps to mouse hemoglobin versus human hemoglobin. So there's a staph virulence factor or a hemoglobin receptor that can grab the hemoglobin from humans, but not mice.
Oh, that's great.
So it's another really lovely illustration of host specificity dictated by this battle for iron.
Oh, wow.
That's pretty cool.
It's an amazing story.
And so that now it turns out that globally there are polymorphisms in humans in transferrin, and they have a lovely map that shows the distributions of these at this position, 589. Not this position, but close to 591, which is we've shown is affecting the interaction with transferrin binding protein. So one variant is called C2. It is present in from 6 to 26% of humans, and there is a change from a proline at 589 to a serine. So it's, as I said, it's 2 amino acids away from 591. Guess what? this variant is very resistant to transferrin-binding protein A from Haemophilus influenzae. Okay, so that's kind of interesting.
Very cool.
What I would like to know is if these individuals have resistance to H. flu, or, he doesn't mention it. And so the question would be, is there any advantage conferred by this polymorphism? I mean, it may have been selected for resistance. But does anyone know? Do you know, Michael?
No, and we're going to begin to lose H. flu as a pathogen because of the effectiveness of the vaccine.
Mm-hmm. So maybe in a few thousand years that polymorphism will be gone.
Well, whoever designed that particular figure, for those of you following along in the science paper, it's figure D, figure 2. And it's the most brilliant figure because it has the map of the globe, and then it has what looks like a clock. And you can see the variation of this polymorphism across the globe so easily. It's the most beautiful figure.
And then they keep that color coding.
Yes, they keep the color coding.
It's very nice. Figures are great in this paper, yeah. So,
but you were making the point about whether some humans now are more susceptible or not, depending on their allele. I'm sure we don't know that yet at the molecular level, but certainly there's good evidence that people can be colonized asymptomatically with H. flu or some of the Neisseria and never have disease, but others, for reasons we don't totally understand, or end up with systemic disease. So it'd be interesting to go back and ask whether a difference in iron or transferrin allele accounts for that.
Yeah. I mean, you can imagine there are other factors as well, right? But this may contribute to it, right? Now, so these two alleles, C2 and C1, they looked at Neisseria gonorrhea binding, so TBPA from Neisseria bound both of these equally. So this doesn't affect Neisseria, but it did affect Haemophilus influenzae. Then they got some clinical isolates, which are available. They're called, these are of Haemophilus influenzae, the Egan isolate and number 11. They both bind C1. Again, that's transferrin, human transferrin, with the proline at 589, but they do not bind C2. Okay, so they are with H. influenzae. These isolates are consistent with what they've seen before. TBPA from Neisseria and both meningitis and gonorrhea bind C1 and C2 similarly. And this strain 15 of Haemophilus influenza, the TBPA, has an intermediate C2 transferrin binding. So I don't know what to make of all these. Clearly there are differences in TBPA binding conferred by these polymorphisms. They say that their conclusion is there's a trade-off between increased binding to C2 versus the breadth of transferrin recognition. But I think the one thing you can come away with is that changes in transferrin will affect TBPA binding. And as we just said, I think the real key next is to find out whether they have any role in modulating the pathogenesis of infection. The last part of the paper, so transferrin has clearly been subject to positive selection. These residues affect binding to TBPA. How about TBPA? So this is the last part of the Red Queen. So the host, the human, has been changing. Does the bacteria change as well, and does it go back and forth? So they look at a bunch of sequences from Haemophilus and Neisseria, and in particular they're looking at TBPA, and they found 10 sites in Neisseria, 9 in Haemophilus that are apparently subject to positive selection, and these are all in the loops of TBPA that bind transferrin. So again, the idea would be transferrin changes, TBPA changes, transferrin, back and forth.
It's beautiful.
It's really nice.
It's the Red Queen.
Red Queen.
Well, or maybe an easier image is imagine two boxers, and one is punching, and then one is bobbing and weaving, and then the other punches and bob and weave, and it's just this dance, bob and weave, bob and weave. Amazing.
Very good. Very good.
They actually have a movie associated with this paper in the supplements, which basically is really cool. It shows you transferrin and the transferrin binding protein, and they bind initially, and then a change arises in the transferrin, so it can't bind. Then the change arises in the binding protein, so they then interact. And as these are happening, the phylogenetic tree is glowing to show you where the changes are occurring. It's pretty cool. If you can see it, check it out. Anyway, basically they find these sites, remember I just mentioned in TBPA, that seem to be under positive selection. They're in the loop of TBPA that bind transferrin. They changed Neisseria TBPA to the Haemophilus sequence at 3 sites, and this reduces binding to human transferrin. So basically, these are sites that can modulate transferrin binding. So it's the Red Queen. The changes are happening on the transferrin and on the transferrin binding protein. Of course, this happens with time, right? When transferrin changes, then the binding protein, then transferrin, and so forth. But the real question is what it means for pathogenesis. So this is great, but in the end, you have to make it real. And so, someone else will probably do those experiments where they try and correlate this with disease in people.
Yeah, actually, that is one thing that Matt, the postdoc who directed this study, is following up on now. He's interested in looking at the bacterial variants and how they've adapted. So it's very cool that he's got a wealth of pathogens and genetics to draw from, in addition to they have in their freezer cell lines from 20 to 25 different primates. Mm-hmm. So whenever anybody in the lab identifies a particular candidate for this evolutionary competition between a host factor, they can go into their freezer, get out cell lines, and then prepare the DNA, and then that sets them up to do the functional tests, the biochemical tests that Matt did here.
It's lovely.
And because they have a systematic approach with these beautiful little dot blots,
right?
I mean, it's cool. So easy to I guess to put it in the simple term is they ask the question, is the Red Queen winning or losing it from the perspective of the microbe or the host?
Yeah, it depends when you look, right?
Yeah.
Sometimes you win, sometimes you lose.
Well, it's back to Michelle's boxing metaphor. Who just got hit?
Yeah. Boy, you're violent, Michele.
Sporty. I'm sporty.
Sporty.
What I loved about this paper is it really brought in evolutionary biology, applied computational biology, but also microbiology, cell biology, genetics, biochemistry. Mm-hmm. Really
structural biology.
Structural biology. Yeah, what a playground for a scientist.
It is. See.
So this is characteristic.
I think there must be other papers like it, but I was really struck by what you just said. I mean, this is a coherence of technology and thinking. It's quite amazing.
There are papers in virology doing this. I think this is the first time they've applied this to bacteria, but they have, as I mentioned at the onset, both Nels and Sarah Sawyer and Harmeet Malik have done this, and Michael Emmerman have done this with viruses and mostly host defense proteins. So this is taking this further. And I can imagine now going after the parasite protein that grabs iron in red blood cells, right? The malaria protein doing the similar thing, and then we'll have
his
paper on
trifecta.
He'll have his trifecta.
Michele and Michael, do you know of other examples of nutritional immunity?
Sure.
That review article is full of them, yeah.
Well, and also the Klebsiella. Mike Bachman has, who's a colleague here at Michigan, has done some work on that, and there's fighting between Klebsiella and lipocalin for iron.
Mm-hmm. I'm talking other than iron.
Other than iron.
It would have to be a microtrace element rather than a trace element, something where you need a lot of it, because most of the other stuff, it would be too hard. And it's really peculiar because how you're asking the question, the siderophores combine other metals that are important for the redox-sensitive dyes. They can go after, they can grab things like cadmium, they can grab vanadium, they can grab tungsten, they can even grab such strange things as plutonium. And in fact, a little bit off topic, if you throw out a rusty nail outside of a house that is downwind from a place that had an atomic open air explosion, you can actually accumulate plutonium on iron nails.
Uh-huh.
So there is a review.
How about other nutritional things? I'm thinking of Boimler's work on the nutrition of salmonella in the gut, where I don't think it's a case like it at all, but
so, Elio, there is a review article called Nutritional Immunity where they talk about iron, manganese, zinc, and copper
Uh-huh.
Okay, so other
Just
other
wondering,
metals.
aside from metals,
you want to keep going.
I want to keep going.
I have some recollection that activated macrophages can restrict tryptophan pools, and that affects some intracellular pathogens.
That's the kind of thing I mean. Yeah.
Yeah.
Good. There must be. I mean,
Yeah.
It can't be restricted to just the rare elements or rare, infrequently found metals. This is just a great strategy, and it must be more general, I think.
Right, because of course all pathogens need nutrients to grow.
Right.
So.
I would look on some of the vitamins that are essential for some of the pathogens. I'll bet we're much more stingy with vitamins we get from our good commensals, and I'll bet you there are pathogens out there that can steal vitamins from both us and the commensals in our gut.
So I had the pleasure of talking with Matt Barber yesterday, who's the postdoc in Niels Eldy's lab, and he's had an interesting life. He went to both middle school and high school in Tokyo, and described it as just a great experience. He and his brother had a lot of independence there, riding the metro and the whole city as a playground, because it's a very safe place. But I asked how he got his start in science, and he said that as a kid, his dad was a golfer, and would take he and his brother out, for hours to the golf course. And his brother took a real interest in golf and became quite a good golfer. But Matt, meanwhile, was kind of digging around in the streams and ponds, mucking around, finding life under the rocks, and eventually went on to study molecular biology as a undergrad at Colgate, and then as a PhD student at Stanford, he learned biochemistry, which we saw played out beautifully in this paper, and then joined Niels' lab for his postdoc. He described one day that was especially exciting in this project, where he had been, he had come up with the idea that transferrin might be a good target for this kind of evolutionary competition, and was studying the sequences and found that only 4 mutations differed between humans and chimps in this one domain of transferrin. And he kind of deduced that there was one amino acid that just looked like a really great target and thought he'd go after it. And Niels said, all right, but if you're just going to make the one mutation, definitely give the human the chimp variant and give the chimp the human variant, and then do your biochemical tests. And voila, he found that he in fact had switched their specificity with just that one amino acid change. So that was a really exciting day, and he knew he was onto something.
Cool.
He also, besides enjoying the great outdoors in Utah, did want to point out that Niels was the originator of something I had not heard about, National Laboratory Mustache Day. Had you guys heard of this?
Nope.
What's that?
So just before Valentine's Day every February, they celebrate National Mustache Day, and they link to it on Matt's webpage. And what I'm seeing is photographs of labs all over the country that have of groups of lab people that have fake or real mustaches. But I guess this got its start because Niels himself has got an impressive rack under his lip. So science is not all serious. They're having some fun as well.
Great. All right. Thank you, Michele.
Mm-hmm.
Nice. I have a few emails to read. First one is from Bailey, who writes, Hey, Vincent, Elio, Michele, and Michael. I'm a first-year microbiology graduate student at UC Berkeley and an avid listener of TWiM. Elio's snippet from the most recent podcast reminded me of a personal anecdote that might be useful to share with listeners. The snippet was about the volatile compounds produced by yeast that attract fruit flies and cause dispersal of the yeast. A couple of months back, I had a small problem with fruit flies in my apartment and decided to try the home remedy of apple cider vinegar and soap to get rid of the flies. The apple cider vinegar is supposed to attract the flies, while the soap breaks the surface tension, causing the flies to become stuck and eventually drown. This concoction helped get rid of some of the fruit flies, but there were still a few buzzing around after a couple of days. That weekend, my roommates and I drank a bottle of wine to help de-stress from the hardships of graduate school. We left the bottle out on the counter with only a couple centimeters of wine left in the bottom, and the next morning it was filled with fruit flies. It seemed that the volatile compounds in the wine were much better at attracting fruit flies than the compounds in the apple cider vinegar.
What would you rather drink, wine or apple cider vinegar?
Well, I wouldn't have left a few centimeters in it.
That's right.
Man, come on. And what's the stress in grad school? I don't know anything about that.
Nope.
Actually, when you think about it, it has happened to all of us that we drink a glass of wine and suddenly there's a fruit fly in it.
Yeah.
Yeah.
It happens with some frequency. That's a great story,
And it's
baby.
the yeast. It's the
It's a
yeast.
great story,
that count
really.
for it.
Yeah, it's the yeast. It's very interesting to hear about the molecular basis for this aroma-based communication between yeast and fruit flies. For any listeners who have come back from spring break or a weekend away and now have to deal with fruit flies, I would recommend using this wine trap. It seems a little bit evil to be luring fruit flies to their death with the scent of a presumed feast, but it works beautifully. Anyways, keep up the good work, guys. Well, if you just have a bottle of wine, it's, whether the flies go in and they drown, but there's no soap in it, right? So it's not this surface tension thing.
They can get out.
Yeah, I'm sure they can get out. It's their own choice.
They go back in.
Can't resist. All right. We have one from John. Dear Twimmers, I can't believe I'm writing in about this, but I have to say I was rather deeply offended at listener Dave's insistence on policing your use of the word Talmudic on TWiM. I'm a religious Jewish scientist. Part of my experience is the understanding that my colleagues who often discuss their Christianity, atheism, Buddhism, Islam, Sikhism, Taoism, Shintoism, and other religious isms are as tolerant and respectful of my Judaism as I am of their beliefs. Science is a human endeavor, and scientists have beliefs and cultures. These do not affect the judgment of a professional scientist, but having diverse cultural perspectives and an uncensored environment is an asset to scientific pursuits. Silencing religious or even formerly religious scientists stifles the pursuit of science. Science should take all comers, provided they pursue unbiased interpretation of data. There's nothing about the word Talmudic which implies you'd pursue anything else. The Talmud is an ancient Jewish cultural artifact. It preserved many of the basic logical techniques of the Hellenistic world and was a building block of the intellectual tradition that gave birth to modern science. My own studies of the Talmud taught me to value the importance of free inquiry and discussion that relies on evidence and logic. There are enough places where Jews face harsh consequences for speaking the words of our culture or sharing our intellectual traditions. I have gravitated to science in part because I feel safe among intellectuals who respect the value of my ancient culture. I enjoy TWiM partly for its Talmudic questions, which remind me of the long, unresolvable arguments I learned as a child. It's a quintessential rehearsal of science, the exploration of questions that we know we will never answer to our satisfaction, but that are explored because of the invaluable lessons that will be revealed for the asking. It makes me smile and feel welcomed every time I hear Dr. Schaechter say, Talmudic question. I would hate to see a fun and enjoyable exercise be marred by the views of a listener who cannot tolerate a single word from a culture that differs from his own. I would never have complained about an atheist question or a madrasa question. I'm appalled that Dave would think it appropriate to scold you in an attempt to police speaking in a way that is informed by your participants' culture. Thanks for all the work you do, and I'm sorry if anything I've said is impertinent. I just couldn't believe what I was hearing when you read Dave's letter.
Gee, John, thank you. I appreciate what you said, and I think you caught the spirit in which we use the word Talmudic. I think that Dave was off the track. It was not meant in any way to imply a religious connotation, even though it may have one, but that's not the way we use it. So, John,
or was it meant to be disrespectful?
It was not meant to be disrespectful. Yeah. I'm sorry if it did. But John said it straight, and I think if you listen to what he just said, that's right on the mark. So thank you again, John.
All right, the next one is from Dennis. Dear Vincent and the Twixers, at some point, this sudden wave of deaths in a small southeastern Nigerian town might become a potential Twix topic, unless it turns out to be poisoned food or water. And he sends a link to a story, which maybe you've heard of a mysterious disease in Nigeria, which has killed quite a few people. I looked it up on ProMed Mail, which is my go-to source for diseases, and it looks like, so this is 18 deaths, and I think, according to ProMed Mail, the WHO said it was methanol poisoning, and This was denied by the Nigerian government, but that's what WHO thinks it is, and so it's not a microbe or anything of that sort, apparently, although we'll see.
Well, we're coming up with Hand Hygiene Day here in a few days, and the WHO has been helping the under-resourced nations of the world develop ways to make alcohol inexpensively. And unfortunately, what they're learning as distributing alcohol is it's much more valuable to sell to drink than it is to use in hand hygiene. So I'm afraid that maybe they were fermenting something and they got methanol poisoning.
Yep. He continues, separately but instigated by this, a thought question occurred. As unlikely as it may sound, are there other vectors besides viruses, microbes, parasites, and poisons from known toxins that could be contagious and which would kill quickly? For example, could someone use CRISPR-Cas9 to modify a common benign bacterium to deliver a toxin? Fanboy of Twix, Dennis. You don't need CRISPR to put a toxin in a bacterium, right? You just put the gene in.
Not most. Yeah, some bacteria maybe are a little more difficult to manipulate genetically, but...
Well,
and that's why we have dual use now as part of our ethical practice of science. Every, ethical individual is supposed to evaluate their work and think about the consequences before they do the experiments.
Dennis, there are a lot of things that people could do, and yes, here and in countries where scientists pursue knowledge honestly, we don't do that. But there are people who would want to do this, and there's not much we can do to stop them. And so that's why we have preparedness programs in many countries to deal with the possibility.
Every institution has a safety board, as Michael was pointing out, where we submit our work to make sure that no one is inadvertently creating a danger.
Right.
Well, there's an argument going on right now, though, about the wisdom of gain of function experiments, where you, on purpose, add what is maybe a virulence factor to something which is otherwise benign. And that, of course, is the people in favor of it point out that that's the way to study them. And that's very true. But, there's a big argument.
Of course, there's also now a discussion about CRISPR-Cas9 and modifying human embryos with it, right?
Mm-hmm.
Oh, boy.
Yeah.
Yeah.
Well, yeah, so, Dennis, I think the Nigerian thing is methanol, and, people can do bad things, but we try not to do it here and in other countries. But someone wants to do it, I don't think it's very easy to stop them. Our last letter is from Anthony, who sends a link to two articles. One is an article about a case of leprosy in New York City. And he's wondering about whether this is common or not. And there has been. So this is a case where the person who got it never traveled anywhere.
That's unusual.
And we do get these in the U.S. They're scattered, and I think they're probably from an animal that has the bacterium in it, right? Don't armadillos have?
You don't have many armadillos in New York City, though.
Who knows?
Their rats are almost as big as armadillos.
We have a lot of things here, but I looked up some other cases in other parts of the U.S. and they implicate armadillos, right? So I don't know. Does anyone else know about armadillos and leprosy here?
They're not in New York. It's it's throughout the Southeast. Yeah. The armadillos got it from eating the pauper graves in New Orleans. Yeah. They went rummaging through and they picked up it from the lepers.
Is that so?
Yeah,
I didn't know that.
And the Hansen facility down outside of New Orleans, out in Louisiana, is where it came from. I'm wondering if Joan Bennett was the one who was telling me that story.
And Hansen disease is another term for
leprosy.
Yeah. He also sent a link to what's called a contagion map. This is a brief history of malaria, leprosy, and smallpox. It's a wonderful world map with some history on it and spread of these infections and so forth. It's quite neat. So we'll put that in the show notes. Check it out. Thank you, Anthony, for sending that. And that will do it for TWiM number 103. You can find it at iTunes and microbeworld.org/TWiM. TWiM and on any of your favorite podcatcher apps like Podcast from Apple and so forth. And if you like what we do, listen. And if you have questions, send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
And thanks for talking with Matthew.
It was a pleasure.
Cool. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, it was a lot of fun.
Ocelloid. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.